I thank you everyone for being here. Can you hear me from the back? Okay, there's two problems. I'm not American, so some people have problems with my accent. I hope that that's okay. It takes the students a few days to get used to it. So if there's something you don't understand, stop me. And then it's also because I'm going to be using maybe the names of some animals that might be unfamiliar to you. What I'm going to try to show you today is some of the research that is going on in my lab. This is a poster that we presented a couple weeks, last week actually, to the undergrads, trying to show them what we do. And what we do is basically research in all those places in the world, working on all different kinds of invertebrates. And I'm just going to give you a little bit of the flavor of the things we do, why we do them. And actually, all these places that I'm going to show you are just places that I visited in the last 2 years, for example. So a lot of our research is very intense in the discovery and exploration of biodiversity. I'm not always traveling, despite what people think. I'm here a long time teaching and doing other activities. So these are some of the projects that I have been mentioned here. One is assembling the tree of life, this acronym ATOL. What it means is trying to infer the relationships among animals. And we had one project for protostomes. Protostomes is a very large part of the animal diversity. We have projects that are specific to the bivalve tree of life, and I'll be talking a little bit about this project. Bivatol, these arachnoid spiders, is a project from NSF that is called PEAT, which is the Partnership for Enhancement of Studies in Taxonomy. So try to get people doing this old-fashioned sciences as taxonomy, revitalizing using new techniques and trying to get new experts that are able to go out there, identify biodiversity, name new species, because there's tons of new species to be described. We have this NeoNeCal, is a project studying the biodiversity of New Caledonia, for example, with some colleagues in France. We have a big project trying to do genomic work. This EST stands for Express Sequence Tags, trying to do transcriptomes of mollusks, and again inferring the relationships among the different groups of mollusks, things like clams and snails and octopuses. And we have very little idea about how they really evolved early in their evolutionary history, how they're related. So those are the types of things that we do. And in general, a lot of the work we do also is try to study biogeography, so the relationships among the land masses where the organisms occur. And we focus a lot on terrestrial arthropods. But you'll see that my work is both marine and terrestrial, and I'll be showing you some of those examples, because my interest is not the ecosystem where animals live, but it's the whole diversity of animal evolution. So why do I study invertebrates? This is just a summary tree for all the animals that we find here. And basically, most of the diversity, about 96% of the animals that we know are invertebrates. And if you look at this tree, our vertebrates will be part of them in that area that they don't let me get to. So that yellow clade, that will contain one lineage, a couple lineages that are just the vertebrates. The rest of the animal diversity is invertebrates. Okay? And in terms of number of known species, more than 96% are invertebrates. If you take into account that probably the rate of undiscovered vertebrates versus the one of undiscovered invertebrates are very different. So we know pretty much every bird, every mammal, a large fraction of reptiles and amphibians. The fishes are not as well known, but you could say that maybe if we're lucky, there's 20% vertebrates to be discovered. While in some groups of invertebrates, there might be 90% of the diversity to be still discovered, okay? Because of their size, because of their where they live, etc. So this is one of the two pictures of vertebrates that I will show you. This is actually diving, looking for invertebrates in the Great Barrier Reef. So a lot of our research takes us all over the world. This is part of this project called Bivatol, where we're trying to use bivalves as a model to understand evolution of animals. And we're doing a pretty deep level work, studying the relationships, the evolution of their anatomy, understanding their genes. And this is work funded by this ATOL program from the National Science Foundation. If you're interested in that program, you can just Google it and you'll find a lot of information. So how do we tackle some of these big projects about trying to understand evolution. Obviously, this is not something that I can do alone here at Harvard, and this is one of our first meetings of the team of PIs, the principal investigators of this program, which are in different institutions, mostly at the Field Museum of Natural History in Chicago, the Paleontological Research Institution in Cornell, and other institutions both in the US, including the Smithsonian or Florida International University, the Citadel, University of Alabama, and also institutions abroad in the Natural History Museum in London or in University of Queensland, for example. So people working together, people with expertise in different disciplines, like one of the guys is the expert in studying the sperm of bivalves, and he's dedicated his entire life to understand how sperm evolves in bivalves, because actually it has a lot of characters that we can use to reconstruct these phylogenies. Other people are expert in studying the ultrastructure of the shell, and others have specialized in dissecting the stomachs of these little animals and just comparing the stomachs among all the different species. So there's a lot of specialization, and then a lot of integration, and then connecting all those things also through DNA studies, which they are based in our lab. So we're the core center for all the molecular studies of this project. And we work together by going together in the field. The different experts need to preserve the same animals, let's say or different specimens from the same species for the different techniques. So we all go together to some trips, not everywhere. This is 2 years ago in Australia, and we're collecting in the different environments where these animals occur, and we're going there as a team and then bringing the animals back into the lab, photographing them to make sure that we have good record of the live habitats of these animals. Many people who have studied mollusks have focused only on the shells, and what we're trying to understand is the rest, the real part of the organisms, not just the pretty shells. So we study them, we photograph them alive. This is just to show you some of the beautiful bivalves. We spend time in boats, dredging from the bottoms of the ocean. In this case, we're doing very shallow water. I will talk later on about some work that we've been doing more in the deep sea. Once we're in the boat, we're just sorting specimens in these trays and selecting the ones that are interesting for our work. We're not always working, you know, the boats have to move from place to place, and then you can use that time to relax. I think that probably was snowing in Boston when we were there in Queensland. I'm sorry. And then we collect all these things. And we went, for example, this trip to Moreton Bay, is a little island near Brisbane. It was very interesting because Australia has relic faunas for many groups of bivalves. And here we have not only these crassatellids, which are very difficult to obtain anywhere else in the world, especially in large quantities because we need several specimens for the different preservation types, but also these things that are growing on top of them is a family Myocamida, which is only found in Australia, and it has a couple species. So we were targeting several species that only occur in that area of tropical Australia. So some of the places we've been sampling are, of course, targeted because we are interested in getting the species that live in those particular places. So that's actually Myocama, one of these bivalves that always grows on top of other mollusks. And it's very interesting also to try to understand their life because they tend to adopt the coloration and the striation pattern of the other mollusks where they live on. So if they lived in a dark mollusk, they will be like blackish and they will have different types of ribs. And it's the same species, so they're somehow adapting the way that they form the shell to mimic the shells that they grow on. These are very small animals that live in the sand, these nucleates. When we look at the shells, they're really boring, but when we start looking at the animals, we see like the food that they use for excavating and how these foods, they only have one, so I don't know if you say they feed from the different ones, will have the different patterns that they're very specific and that we can also use to understand these animals. You can see by transparency some of the parts, internal parts. You could see the coil guts or the gills that they use for respiration. This is another very interesting bivalve. It's one of the very few bivalves which is a carnivore. So most bivalves are filter feeders. Some of them sediment feeders. They have these little tentacles. They get particles from the sun. Most of them filter feed water through these inhaling canals, and they go through the gills and they separate the food particles from the water. But these guys have developed this siphon. They live in the deep sea where there's nothing to filter really. There's very few particles in the water. And what they have developed is this system that they can basically suck little arthropods that are swimming around so they can hunt for little copepods or ostracods or things like that. So it's one of the lineages of carnivore, or the only lineage of carnivore bivalves. And we're very interested in understanding how these evolved, for example. These are tiny limopsids. These are about 1/10 of an inch, but when you see these things on the microscope, they have like nice structures in the shells. This is the periostracum that covers them and protects them from even parasites or other things. Neotrigonia, this is an amazing animal. I'm sure you don't know these bivalves. These were very common bivalves in the Ordovician, and they went extinct. It was an entire lineage that went extinct, except for 6 species that live only around Australia. But they're very important evolutionarily because all the large, pearly, freshwater bivalves that you might know, they're actually sister group to these things. So these are the remnants of the marine ancestors of these big radiation of freshwater bivalves, and now are in all the continents. But the marine species, which were very widespread, they're just pretty much extinct, and there's only these 6 species. So we wanted to get some of those, and we're now trying to get the entire set of genes that are like the transcriptome, not the whole genome, but at least all the genes that are transcribed that have a function. So this is one of the model bivalves we're using now. Observations of their biology are very important during these field trips. It's not that we only fix the animals for taking them in the lab. You have here an animal that was disturbed. It was in an aquarium. The water was getting warm. And what many animals do when they get stressed, they spawn. They want to just pass to the next generation. But this was very interesting because it's a hermaphrodite bivalve. And if you see, this is the sperm that is coming out and the eggs that are coming out of the siphon. So it's already releasing the sperm and the eggs. And we were able to get some fertilized embryos from these animals. And that's another of the families that is only found in Australia, for example. Beautiful Phrenomia, the shell is so thin that it breaks when you touch it. So you can see that the shell is actually damaged here. It's impossible almost to catch these animals with the entire shells, and another interesting one. So we bring those animals in the lab, we distribute the job. We have there on the right John Healy dissecting the males to see if they're ripe to get the sperm. Alejandra was a technician in my lab. She was an undergrad here, and then she was entering all the data from the collecting sites, and we spend a lot of hours actually working in the lab. That's the thing, when you're doing field work, there's no time for anything else than just your many hours in the field that you can get to the lab, and if you don't sleep, you don't sleep. You need to process the animals, fix them, photograph them, document everything that you've done. So it's hard work, and that's the setup that I use for photographing many of these animals that you saw. They're not photographed, most of them in situ, but we photograph them in the lab, and you have to wait until they start doing things. So that's our project on the bivalves. And also, not only when we're in the field, we get together every few months to discuss the project. This was a meeting we had here in December 2008 to then also discuss all the data that we have generated under the ground, to discuss how these morphological characters are evolving, and try to compile them into data matrices that we use for analyzing them, and try to then come up with these trees that are reflecting the relationship of the animals, these phylogenetic trees, which is what we do in the lab. So we also apply a lot of field work. It's very important for my research. I couldn't do any of my research if I didn't go on the field all the time and get new animals. So we want to teach the students also the importance of field work. And this is where Peter came with us in one of these years to this trip. This every spring break, because the university doesn't allow me to take students, even if it's for work during the normal lecture days. So during the spring break, we take the students of the Invertebrates course, which is OEB 51, to the field and learn a lot of these techniques. And the students learn, many of them, is the first time they've been in the sea, it's the first time they see a lot of these animals, and it's a really eye-opening experience for understanding biodiversity, for appreciating biodiversity and the research that goes on with these animals. So This was the class from 2 years ago, actually. We go to this beautiful station in Bocas del Toro in Panama. It's a Smithsonian station, so the relationship with them is very good. We have good facilities for doing work in the labs and good microscopes, and most essential, a beautiful place with warm water, which is very important if you want to keep people in the water for many hours. I mean, seriously. When I was a student, they took us to colder places, and it's not fun. Well, it's fun, but then you get really cold. So here you can spend a lot of time in the water with boats. They all snorkel. Sometimes we've done some diving with some of the students that were certified. Normally we don't. And it's very important to make sure everyone is safe, make sure they know the animals they shouldn't touch, even though they're going to touch them anyhow. But then they can't blame you that you didn't tell them. And then we spend a lot of time underwater, and the students are appreciating all this biodiversity. And it's very different for us to see these things dissected in the lab here, or even in our labs we have live specimens that we get shipped from Florida, but they have no idea about what part of the ecosystem, how many sponges are there, because you see like 10 sponges in the lab, 10 cnidarians or a number, whatever, but you have no idea that really all these ecosystems are basically cnidarians and sponges, and then all the other animals that we might know better, like the different worms and mollusks, they're actually much harder to find. They're a very small fraction of the total biomass of these ecosystems. And they also learn to appreciate the importance of preserving reefs and preserving all these areas where we can actually host that biodiversity. We search in all the different environments, not only in the reefs, also in the mangroves, and they see that those are very important for the reproduction of fishes, for example. Many of the big fish go there to spawn in the mangroves because they're protected in the roots, and you have a lot of the schools of the smaller fish around there. They see all the different species that live in those places. This is one of the species they shouldn't touch. This is the fire coral, and yeah, it's not that bad, but the name is worse than other things. And as I said, sometimes we do some of the diving. This was actually with one of my grad students because we wanted to get some of the deeper specimens to bring and show to the students. But they really learn also to be in the water, to be in the environment. We collect the specimens that we need to bring them into the lab. Then we return many of them, if they haven't died yet, to the place where they came from. And we don't collect the corals and things like that. The corals, we have already the samples, and that's what. And also we do a lot of this documentation, and these are many of the typical animals that they will see, and things like these ribbon worms or nemertians. This is a flamingo tongue snail, which is very common living in gorgonians, or that's one of the most typical worms that lives in a tube. Once you relax it and put it outside of the tube, and you can start learning their anatomy and appreciating also their beauty. It's a tropical place, so you get big things. This is one of my grad students was showing off with he used to work on beetles, now works on other things. So I think that bringing this part of the biodiversity exploration to the students is also very important. And this is part of why I am who I am, because I did these things when I was a student, because I learned those things, and I really decided that that's the career that I wanted to take. Things can be sometimes, small teams and easy to organize, and other times can be like really difficult, large-scale projects. And this is one that we had about a year ago. We were at sea in this large vessel from the Spanish government. We went to explore a very interesting site in a drop-off in the northwest side of the Iberian Peninsula of Vigo. And what we were exploring is this subaquatic mountain. So this is an area that comes from 5,000 meters depth, so about 15,000 feet, up to this mountain, which probably that mountain might have been outside the sea level, above sea level at some point, but it's now basically a plain that it reaches about 1,500 meters, about 4,500 feet. And we were sampling in many of these white spots. These are spots that have been sampled. We didn't do all these in the last campaign because this takes a lot of time. But it's also very interesting to work with large groups of people with very strict shifts of, every 6 hours you're on shift. Every we had 3 shifts of 6 hours each. So you had 6 hours, then 12 hours nothing, then 6 hours, then 12 hours nothing. So you can see how some shifts were at night, some were at day. You want to keep these large operations working 24 hours a day because, first of all, it costs a lot of money to put a ship like that in the water, have so many people there during 1 month. And some of the operations that we were doing, like just bringing a net down to 5,000 meters and then back up, the whole operation might take more than 10 hours, for example. So these things are logistically much more difficult than some of the other work that I had done. We had to try these suits in case, big disaster happens. So these are evacuation suits that you can be floating in the water for many hours. Vitu Gorgori was the chief of the campaign. So in these cases, there are very clear hierarchies, like the army a little bit. There's one person who has to make decisions. That person needs to make the decisions, cannot say, well, whatever you decide. It's very, very strict. And then we had our teams, and this was our team. So this is our team that we were operating every certain number of hours. It was mostly Spanish, but there were some international colleagues, like Enrico Schwab is an expert in chitons from Germany. And then I was invited from here, even though many of these guys are colleagues from when I was in Spain also. This was our deepest point. 5,200 meters, that's 15,000 feet. And getting down there, it's really hard. And I'll show you some of the things that happen. We work with many different types of gears. This is an epibenthic sledge. What the intention of this one is that it's being dredged in the bottom. It doesn't go below the sediment. It's just lifting some sediment. And the sediment and things that come from the sediment end up in these 2 bags there, and then you bring it up and recover things. So these are for things that live on top of the sediment, like, for example, little amphipods and things like that. So we're not going deep into the sediment, but we use all different kinds of different gears that will go in the sediment, or box corers that will just get to the bottom and then kind of just shock the bottom, get a sample, and then bring it up. And that one is for more quantifying, because you know exactly the area and the depth that you're penetrating into the sediment. So you can see how different types of studies might require different types of gears. And again, many hours in the microscopes inside of the boat. Now the microscopes are moving, your samples are rocking. One day it got really, really, really bad. That day we didn't do any microscope and just tried to survive for the day. We also bring these bottles for people studying larvae. So these go to the bottom, and at whatever depth you want, you open the bottles, the water that is there comes in, and then the chemists and the larval biologists get their samples from the water from a specific depth. This is our bulldozer. This is a little cylinder, like really thick, like the walls are this thick. And the intention of this is getting to the bottom and breaking the rocks and then bringing samples of the rocks. There were geologists very interested in seeing whether the bottom was granite or something else. And also some of the animals, very tiny animals that we collect, they're always attached to the rocks. So unless you get pieces of rock, you're never going to get those animals. And so some of this is the one that I show you. You see the big chain and that thing. It's just designed to break rocks. These are some of the ones that get to the bottom also and just get the sediment for quantification. This one didn't work so well, so we ended up using one with a single one. And this is a very typical dredge that is just like being dredged in the bottom. But since you don't know what's there, and it might take several hours to go down, and many times if they get caught on rocks or something like that, they will break the net, and then the sample comes up empty. So they redesign it with these like very thick leather layers on each side, so the intention is the leather is protecting the net from inside. So there's a lot of logistics and things that you need to be readapting, and every failure could be sometimes 12 hours of redoing the work. This is the Agassiz troll from our Agassiz here. That's what he used for collecting a lot of the invertebrates. You can't really send this too deep. We went with this one down to 1,000 meters. It's designed to collect larger invertebrates, things like big sea stars or things that are on top of the sediments. And this was our big box corer that was made like of tubes that are about this thick, but they were hollow. And the designer forgot to drill a little hole. And what happens is that when these things go very deep, the difference in pressure is brutal. So what you see here is actually this tube like that was compressed completely, and I'll show you just a detail of it. This machine was useless after that. And what happens is, you can see here the tube, how it was before it went down to 3,000 meters and how it came back. And we do the typical thing that often in schools they do also. They bring these styrofoam cups, and then when you bring them down, you paint them, and when you bring them down, they're like much smaller than they were originally. And that's all for the pressure. People love to send also like these styrofoam heads of mannequins and things like that, and then you have your little reduced head. So pressure is, is very big there. So this is going to condition the type of gear you can send and how you do things. And also you have to think that a machine like that might be super heavy. When it's working at shallow depths, it's great. You drop that thing 300 feet, it touches the bottom and it's fine. But when you're dropping it like 5,000 meters and you need to have a lot of wire going in, I mean, the weight of the wire ends up being much more than the weight of the device per se. So if there's currents underneath, it might just like be lifting it. So it's hard. It's very hard to work at those depths. And for me, this campaign was very important to learn some of those techniques. I had done a lot of marine work, but never deep sea. So I really enjoyed it very much. And again, we were photographing a lot of the specimens. I was designated the official photographer for these things. And you get really rare deep sea animals. Some of them don't look very appropriate to show in some places. Large pycnogonids, and then all sorts of deep sea mollusks. And these actually I photographed because they came from 5,000 meters, and this is one of the areas where there's very little knowledge about large mollusks in that. And that's a pretty large protobrand. We still don't know what it is. And here's one of the night ships. So it was really fun, but it was really tiring because in your ship you're working the 6 hours, and then when you're done with your ship, you still want to see what other people are bringing up. And I was photographing and then preserving my animals. So during your ship, you do all the common tasks for everyone. And then if you have work to do for yourself, is that. So very few hours of sleep during 15 days, except in the transits and things like that. So now we're going to move to land. And a lot of the work, and I would say this is even more important work in my lab in terms of productivity, is work on biogeography and phylogeny of all sorts of terrestrial animals. This is my pet animal, these really tiny daddy long legs who nobody knows, who nobody cares about, but they're great models for studying evolutionary biology. This is what we do, studying evolutionary biology. And these things are really interesting because we know they've been evolving for about 400 million years. And that's much more than any vertebrate that you know, much more than insects weren't around at that time. So these animals were on land about 400 million years ago. They live on all continental land masses. Of course, they've gone extinct from Antarctica because it's too cold now. But they're all the land masses. They've been around for 400 million years, and they're not in any oceanic islands, which means islands that have originated in the middle of the ocean from volcanoes or coral reefs or things like that, which means that these animals are not dispersing through water. Right? So if they're present in all the land masses, it's because they were there once all the land masses were together configuring the previous continents or supercontinents or megacontinents like Pangaea and Gondwana and things like that. So this is a current phylogenetic tree of representative species there. And what we have done is color these trees by our taxonomy, what we call families, and see where those families occur. So here in yellow, we have representatives for what we call the circumantarctic Gondwana. So New Zealand, southern South America, Australia, southern Africa. What is the temperate Gondwana? In green, for example, we have a group that is found only in tropical South America and tropical West Africa. And you know that those two continents were once together. Here in orange, we have all these things that are in Laurasia, North America, Europe, all the way to Japan. So you can see that there's a lot of geographic structure here that makes sense with our paleo reconstructions. And that's the type of work that we do. We're trying to find different groups that live in the different land masses, and then we can go back in time and try to reconstruct the distributions of those organisms according to the maps, so to the paleo maps. And we can even go back in time to the Jurassic and understand why these two families are found in West Africa and Northern South America, why these ones that are around Antarctica, they were all a continuous group of species. And you can go back to when the whole Gondwana and Laurasia was together in this big Pangea. Okay? So a lot of the work we do is going to all these places, because we need to have representatives from all these different land masses, understanding their evolution, their phylogeny, and we use often DNA or morphology for that, and then try to date those phylogenies to see whether the occurrence of those things in two land masses precedes the separation of those land masses or not. So that way we can explain whether these two things occur across the Atlantic because they have dispersed through the Atlantic, or because they were there before the Atlantic opened up. And that's a lot of the work we do. And with that, we need to do field work in many places. Like last summer, we were in Cameroon and Gabon collecting some really interesting animals. And it's great to work in these places and get to know people from different areas. We were in this tiny village of Ototomo, and they were fascinated with the things that we were doing there, and everyone was trying to help us. The problem is that they were bringing you animals that are not the ones you want, and you have to be nice. Oh yeah, that's great, but it's not really that one. So that happens a lot, and everyone has seen your animals always. Okay, you go anywhere, you say, yeah, I'm looking for this little thing that nobody has studied in hundreds of or many years. And everyone will say, oh yeah, I have them in my bathroom. I have them, yeah, oh sure, show me one. Never. And we had a few targets there, some specific animals for our biogeographic studies. These two animals on top are the juvenile and the adult of a very cool arachnid. So it's related to spiders and scorpions, but it's very different from them. They're called Ricciolae, and they're again only found from Mexico to tropical South America and West Africa. And we had a lot of samples from this side of the Atlantic, and we wanted to get more samples from the other side. This is again your house centipede, except that this is not the common one, Scutigera coleoptrata, that you can find in your basement or even in this building. This is Sphendononyma, which is only found in Brazil and West Africa. And we are working on the evolution of these groups, and we needed to get some of those. In the bottom right, you have another type of arachnid that if I gave it to you, most people will not recognize it as an arachnid, will think that it's an insect or something like that. It's related to the vinegaroons. I don't know if you've heard of the vinegaroons. There's one on display in the museum. These, they call them also whip scorpions, but so they're related to these things, very tiny, and we're also interested in understanding that. My group, the one that I do most of my work, are daddy longlegs, Opiliones, and all sorts of really weird daddy longlegs that I had never seen, including this thing. This is what really drove me. This and another animal is what really drove me to that part of the world. I already went to Equatorial Guinea a couple of years ago, and I failed to collect that animal, and I don't like to fail to collect an animal, so I had to go back and find another species from the same genus. This time in Cameroon is a species that I had described based on museum material. I had more information about where it occurred, and it was that village of Ototomo, and we finally found it. It was the last family of that nice phylogeny that I showed you at the beginning, so it was the last family that I was missing. And now we have it, so that's good. And there's a lot of other cool things that we study or look at or photograph, and there are great diversity in these places. We're doing a lot of work on myriapods, mostly centipedes, not so much millipedes, but I'm also interested in them because they're very good models for biogeographic studies. These things don't really move around that much, so that's very good. And yeah, we photograph some vertebrates sometimes when we see them. People like to see them, but this is the other animal that took me there, Mesoperipatus tolonii. This is a velvet worm, and velvet worms are very charismatic. We're working on velvet worms in the lab now. It's a family that was distributed across Gondwana. They can be very common in places like South Africa, New Zealand, Australia, and even Central America, but there's only one species in tropical Africa. So there's several species in South Africa from a different family. In all tropical Africa, there's one species. It was collected for the last time about 100 years, This is the only 3 papers that we have on them. Paper from 1898 of the guy that discovered it, and then a paper a few years, 6 years later from himself. This is a Frenchman, and then from a German in 1926 that studied the nervous system of this animal. Nothing else has been done with new material from these things. We wanted to get material for studying their DNA, to plot it into this big phylogeny of the group, and we went there, spent a month in Cameroon and Gabon, and the last day of the expedition we found 1 specimen, which is enough, but we were beginning to get a little anxious. And that's the thing of field work. Sometimes you really need to optimize things to find the animals. This is another species. This one is from New Zealand. They're beautiful. They're really beautiful animals. If you Google velvet worms or peripatus, you'll see them hunting. There's a couple of videos, one from the BBC and another one from I think a guy from, I don't know where is he from, but in Central America, where you have these things, they just have these glands in the mouth that just secrete this glue, and they hunt the insects with this glue. It's just beautiful to watch. The insect doesn't like it so much. And this is the distribution. So we are doing this, again, these worldwide studies. We're trying to date the separation of those continents based on understanding the evolution of these very charismatic animals. And they're rare to find. And this is the current sampling we have. A lot of species from this red family. This is the circumantarctic equivalent to those daddy longlegs. They have the same distribution. And this blue one is equivalent to that group that lived in tropical South America and West Africa. They're also found here in Southeast Asia, which is also part of the pantropics. So we find this, again, this geographic structure, we can recover it from studying their morphology or their DNA sequences. And that's how we want to match those things and try to understand geological processes also by looking at some of these animals. Yes?
How many species of oncophorans are known?
There are about 100, not so many. There are about 100 known. Yeah. Many more in the red family than in the other one. This family is Peripatopsidae, this is Peripatidae, not very different.
Does this mean they're not in North America?
There's no in North America, not now. There's the group is actually very interesting because there's ancestors in the Cambrian that were marine. And I don't know if you might have heard about things like hallucigenia. So they were the ancestors of these onychophorans, probably. So. And I will finish now with this last one because it's very different to what I do normally. I go to tropical places and the ocean. And this summer we had to go to Greenland in July. In Greenland it's very different. This is the Arctic station from the Danes that they have there. Beautiful places. I wasn't expecting it to be so beautiful. There's a lot of icebergs everywhere. And we were going to this specific homeothermal spring, only one, where we could find an animal that was discovered in the year 2000, and that is a new phylum. It's equivalent to vertebrates, or equivalent to mollusks, or equivalent to annelids, but it has just one species known. And we wanted to do the whole genome of these things, and we were sampling just the mosses that are in this spring. This one, 100 meters away, it's not there. And with those mosses, then we will squish them in the lab and just study them and find this tiny animal about 125 microns in size. This is about an eighth of a millimeter, so that's about a 20th of an inch, or something like that. So really tiny animals. We needed to get a lot of them because we want to do the entire genome of these things. We need to get enough DNA and RNA to do the type of work. So we want a big team of people, including the people the person that discovered the animal, Reinhard Christensen, who has discovered a lot of these tiny animals. Yeah, it's icebergs everywhere there. We went in the water. We swam in the water, very briefly, but we did. And he's a god of discovering new animals. He has discovered 3 animal phyla nobody else has done. Some really small animals that live in very strange environments. One of them, an animal that lives in the mouth parts of our lobsters here, and the cycleophrans. So he is also the one who discovered those animals. And there is Reinhard in his homeothermal spring, really happy every time that he goes there to look for Lemnognathia. So that's, you know, the weather, you might see icebergs there. When the sun is out, it actually might be very warm. Wasn't expecting that. Apparently it's bad when it's too warm because there's a lot of mosquitoes. But it wasn't even that bad with mosquitoes this year. And just to conclude, I mean, a lot of the work that we do, I've shown you the nice part, but we do a lot of work in the lab. And some of the things we're trying to do is assemble these entire genomes or entire transcriptomes. And these are some of the graphs, for example, of, you know, now the new generation sequencing technologies that we're using, where we can generate hundreds of thousands of sequences for these animals. And they try to reconstruct. These will be like the distribution of genes that we have longer than, for example, this is 2,000 base pairs, and we have a good distribution of genes that we can accumulate. This is actually 454. What we did last year is, you know, now in-house we have these new sequencing machines where this graph will give us many more sequences of much greater length, for example. And that's what we're doing right now. We have some samples running in the machine. So with that, I'll just say goodbye. And if there's any questions, just feel free to ask. Yes, there's another one there, since you asked one already. Yeah?
Yeah, I had a question about your cladograms. What are you using for statistical techniques? Because they vary all over the place for what you want to do to get a decent cladogram. And what basic data are you using? Are you using morphology? Are you using DNA? Are you putting it all together and using those to collect?
Well, it depends on the study. So in some of the studies with a few genes and morphology, we might have enough information to resolve things pretty confidently. When we go back to study very old processes like the origins of all animal diversity or the origins of mollusks, that's when we've moved into these genomics. So in some of our, that first tree that I show you is based on data from 1,500 genes for about 100 taxa. Our next analysis probably will contain information in maybe 5,000 to 10,000 genes. And that's how we're doing it. And then, of course, there are taxa. There are some organisms that are very unstable because they might have very unusual genomes, also very unusual morphology. And you know, we'll keep trying, adding more diversity there, see if we can nail down those last ones. But the reality is that now we have a much more stable tree than the ones we had just 5 years ago.
And is the TOL project using the same statistical techniques for all the cladograms that they produce?
No, there's different flavors of techniques for doing cladograms. Different researchers might have preferences for different ones. But now most of the results that are emphasized will appear with all the different techniques. Yes. And then you will have.
Will this new phylum be published, and does it go back to the Cambrian explosion?
Well, we don't know. The idea is that all the phyla originated in the Cambrian, but the reality is that this new phylum is nested within a clade that we have no fossils. So many of the other phyla that are related to them are also small, meiofaunal. Meiofaunal are things that live in between the sand grains, and we have no fossil record whatsoever. So well, we can infer that all the major lineages of animals originated in the Cambrian because we have fossils. For these ones, we need to use indirect techniques, which will be dating these molecular trees or things like that. And we infer that yes, they might have been originated in the Cambrian, but the evidence is a little bit more sketchy. Maybe the whole clade originated in the Cambrian, but we don't know if the phylum originated then.
Which clade does it nest in? And when is
It's
it
something that we call platyzoa. It includes things like platyhelminths, and there's no old fossils of platyhelminths, flatworms and things like that. And then other smaller things like rotifers, you know, rotifers, the wheel animals, and gnathostomulids. Those ones probably you don't know. They're also very small. They have very large jaws, the same as these micrognathozoans. So, and we have no evidence of fossil record for these things, of all fossil record.
So how well did the critters that you're bringing up from really deep down, how well do they survive the trip?
Yeah, most of them are fine. The ones that have problems will be fishes, for example, because they have the bladders or things like that, and there's typical pictures of a fish with the eyes coming out of the socket. That's because they have, yeah, I mean, it happens. You know, that you bring them from 5,000 meters depth, and then when they're being decompressed as they come up, so any air they have inside of any cavity in the body will implode, will explode. But things like little things like mollusks, they don't have large cavities. They don't have any type of air inside, so they're fine. Most of the invertebrates will be fine. They will be alive at least for a little time. The problem is more they change in the temperature of the water sometimes than just the difference in pressure. And you're bringing them from temperatures in the bottom will be around 2 Celsius uniformly, and then you bring them up to maybe 15, 20 degrees water as soon as they're outside. If this was in November, it was like really warm there. So you need to keep them refrigerated actually to keep them alive. So that's more of a problem. Okay.
Yeah.
Is there much controversy about where to place a new organism in the phylogeny?
So sometimes there is, yeah, and there are organisms that are more controversial than others. And I could go over a list of some of the animals that are really weird that we have placed traditionally in one place, and they've jumped to another one with one type of data and to another place with another type of data. But those are animals normally that, as he was asking before, they might be more uncertain with the different methods of reconstruction, but there are some of those.
So do you work that out when you have your group meetings with your...
Yeah, those are the things that we want to solve.
Yeah. Do you use fossils at all?
We do when we combine with morphological data. If we have fossils that have intermediate characters between different phyla and they're well preserved, one of our team members is a very good paleontologist, and we code the morphology for those fossils whenever it's informative to some of these problems. In the past, when we use a few genes and morphology combined, that probably was able to change the topologies a little bit. The problem is that now when we combine a relatively small morphological matrix with even a few hundred characters with, let's say, information from 5,000 genes, that information gets so diluted that the only thing that it really does is to anchor the fossil in the place of the tree that it wants to go. But then for us it's good because we can then use those points also for trying to calibrate our phylogeny. Since we have a fossil with an accurate placement in the phylogeny with an exact age for the occurrence of that taxon, we can use that later on for calibrating our tree and inferring that the separation of these two lineages occurred 350 million years ago or something like that. Yeah.
The individual, and I can't remember his name, who discovered the organism in Greenland, what were the circumstances of his finding this?
Well, that was exploring biodiversity. He was taking his class to study Arctic biodiversity, and they were looking for small animals, meiofauna. He's an expert in meiofauna, and looking at meiofauna in different places in the ocean. Now, this is fresh water. So they needed to set a camp. And what they wanted to do is, before you set a camp, is where do you have water that you can drink? So they took this water, and they were looking at the water to see what type of animals lived in that water. That might be good indicators for good quality water or bad quality water. And they found these things there. So, yeah, it was a coincidence. But coincidences don't occur to somebody who doesn't go there and is going to a remote place and is trying to look for drinkable water, trying to discover weird animals.
Favors have prepared for him.
There's always the chance of opportunity. And, there's another one, like this other animal he and his student discovered, the one that lived in the mouth parts of the lobsters. There was another similar story. So the student, Peter Funk, went to Roscoff, which is a very important marine station in France. And he was looking for rotifers, the wheel animals that live in the marine sediments. And he was washing his samples through running seawater system that they had in the lab. And what happened is that the running seawater system had been contaminated with rotifers. So all the samples he had collected in the summer had the same rotifers. He was trying to establish the differences between the different types of sediments. So he went back after a whole summer of work, went back to Copenhagen and talked to this other professor who's a very famous biologist, Klaus Nielsen, and told him, look what happened to me. And he's like, well, I had this other wheel animal, this other rotifer already here prepared for the transmission electron microscope. It's really weird. Why don't you study it? And then he studied that one, but it wasn't another wheel animal. It was this new phylum that is actually not really related to those wheel animals. So he thought it was a commensal rotifer that lived in the mouth parts of the lobsters, and it wasn't. It was something different. So, many of us might see new things and not appreciate them. And I think it's after a lot of perseverance. And then they spend a lot of time trying to figure out the life cycle and discover all these different larvae that they have. And what they thought it was a wheel animal, it was only the female feeding stage. But then that one generates these larvae and these dwarf males. And it's a really complicated life cycle in that case. So they spend years studying them. So, yeah.
Biodiversity in the Amazon, this is one of the things that we always told our students, that the biodiversity in the Amazon is unlike anywhere else. Have you done a lot of work in the Amazon?
I have done work in the Amazon, yeah. And it is true that it's a very large biodiversity in the Amazon, but I don't think it's unique. What is unique from the Amazon is the extension of that forest. But in Southeast Asia, in Indonesia, you have also places that I'm sure that match the Amazon in the amount of biodiversity. Perhaps they're less studied, and the problem is that we're losing those really fast for the logging companies. There's many places with a very large biodiversity. And I think the difference will be also the bias. In places like the Amazon, most of the biodiversity is going to be in arthropods, which, they're 85% of the total animal diversity. But you have low diversity in other groups of organisms. If you go to a coral reef, what you're going to have is not as many species as in the Amazon, but you're going to have a huge representation of all the different animal phyla. I think that most animal phyla live only in the sea. Only a few made it into fresh water, and even fewer made it into land. But you find pretty much everything in the sea. The one thing that is unique about the velvet worms is that the only animal phylum that right now is exclusively terrestrial. All the other ones have members that live in the sea and in fresh water. And that one is the only terrestrial, strictly terrestrial one.
So is that basically your most interesting of the animals?
I'm interested in all of them. And I might have interest in different ones through the day. In the morning I'm working. I love them all. I could never make a choice. I think that's why I ended up working on all of them. Yeah. And I have people in the lab working on all these different projects. All of these projects and other projects are currently ongoing in the lab.
Yeah.
Have you found any new classes or families of proteins that are unique to a farm that have never been?
Yeah, I'm not doing that type of work, but there's people like Craig Venter, who you might know. He's the guy that did the alternative sequencing of the human genome, the private one. And what they were doing is actually going in the ocean and just collecting these picoplankton, things that we don't even see, and sequencing it. And they discover new families of proteins that we had no idea what they did. So, biodiversity, we need exploration in all kinds of things. I'm more interested in the organismic part, but some people might be interested in molecules and you name it. Yeah. There was
I was just going to ask, are there any velvet worms on volcanic islands? Do they disperse through the ocean or not?
Not so much, no. They're not good dispersers. And so they are only in these continental landmasses. So they're in some islands like Tasmania, but Tasmania has a continental origin. New Zealand has a continental origin. But they're not known. Well, no, I'm lying. There's one known from Galapagos, but it's a known introduction. So it was a man-mediated introduction.
And have people done the phylogeny of like the ones on Tasmania, New Zealand, where they came?
We're doing it now. Yeah, and we have these several instances of TransTasman Connection. So a couple different clades that have sister groups in Tasmania and New Zealand. And what we're testing now is whether that divergence, those two instances of divergences, they have to be older than the separation of Tasmania, New Zealand, and Australia. And that's what we're actually testing. If the test, if we're not correct, if they're younger than 80 million years, then our whole hypothesis that these things don't disperse through ocean is killed.
What is your view on the multicellular organisms developing from sponges? That was one of the theories put forth about the development.
Yeah, they don't develop from sponges. They share an ancestor with sponges, all the other animals. If sponges are the most primitive animals, which, there's debate now, but if they were, you know, it doesn't say that they came from sponges, that the sponges and the rest of the animals share a common ancestor. It could have looked like the sponge, or it could have looked like another thing. So that's a little bit difficult sometimes when we're talking about phylogeny and evolution. You know, things we can never infer if, you know, we find a fossil in the fossil record. We can never say this animal came from that fossil. You know, we can say, our techniques allow us to say that that fossil and this other animal share a common ancestor. But we don't know whether the common ancestor looked at the fossil or like the other thing. Okay. So, but normally, organisms that have more primitive characters, we tend to talk about them and assume that they are, you know, the ancestor. But there's no way we can infer that.
Thank you.
All right, thanks for coming.